Hi viewers, in this video we will get a quick overview on production of tetracycline. We have already studied the production of penicillin and streptomycin. Now we are going to study about tetracycline, its uses, mechanism of action and types, then structure, production process, and finally recovery. Tetracycline. Tetracyclines are chemical substances produced by a microorganism that are able to kill other microorganisms. It does not cause any toxic effect to the person, animal, or plant. It was discovered by Benjamin in 1945. Tetracyclines were derived from Streptomyces species. There are three types of tetracyclines: short-acting tetracyclines, intermediate-acting tetracyclines, and long-acting tetracyclines. Tetracyclines are broad-spectrum antibiotics, and they are effective against gram-positive and gram-negative bacteria. It can be prepared microbiologically as well as chemically, and it has low toxicity and it requires high dose. The basic structure of tetracycline is given here, and the structure composed of a naphthazine ring, that is a 4-ring structure. Then they are yellow in color. Okay, please note the structure. Next we can discuss the uses of tetracycline. Tetracycline have antiviral, anti-cancer and antibacterial activity. It is used to treat respiratory tract infection, cholera, urinary tract infection, salmonellosis, shigellosis, typhoid fever, brucellosis, etc. And also it is used in the treatment of human and veterinary diseases. It is used for the preservation of fish, meat and poultry products. Let's move on to mechanism of action. Tetracycline inhibits protein synthesis by blocking the attachment of charged amino acyl tRNA to the A site on ribosome. That is, tetracycline binds to the 30S subunit of the microbial ribosome. It prevents the introduction of new amino acid to the peptide chain. So this is the mechanism of action of tetracycline. Similarly, we can say tetracycline inhibits the protein synthesis or translation process. There are different types of tetracyclines: tetracycline, doxycycline, minocycline, oxytetracycline, chlortetracycline. Next is the organism used for the production process. The mutant strain of the Streptomyces aureofaciens are used for the production of tetracycline. Streptomyces aureus, Streptomyces flavus, Streptomyces antibioticus, Streptomyces cellulose, etc. are used for the production of other tetracyclines. So the choice of organism is dependent upon the type of tetracycline. Inoculum preparation uses several stages. Spores dried on sand or lyophilized sample is used. Different streptomyces produces different tetracyclines. Okay. Next is the production of tetracycline. Production of tetracycline is very sensitive to chloride content in the medium. It leads to the production of chlor-tetracycline rather than tetracycline. Production of chlor-tetracycline is simpler than other tetracyclines. The fermentation medium consists of cornstarch liquor, soy flour, peanut meal, carbon source, etc. The production process involves several steps. First step, inoculum preparation. Then the inoculum is transferred onto the agar plate. Then it is transferred to the shaker flask. Then to the fermenter. Okay. Then the fermentation process is carried out. After the fermentation process, the mycelium is removed. Then carry recovery and purification process. Okay. First step, inoculum preparation. The organism comes in the form of lyophilized spores. The inoculum preparation requires several stages. Lyophilized spores or dried spores on sand are used. Then the spores are inoculated to inoculum tank. Okay. Then the tank is incubated at 28 degrees Celsius for 24 hours. After the inoculum preparation, 5% of the inoculum is added to the production fermenter. The fermentation runs for 60 to 65 hours at 28 degrees Celsius. Okay. The pH range is about 5.8 to 6. Aeration and agitation is also required. Agitation is with mechanical agitators. It is a submerged fermentation requiring 0.5 volume per minute aeration. Here, lard oil is used as the antifoam agent. So the fermentation temperature is 28 degrees Celsius, pH 5.8 to 6, time requirement 60 to 65 hours, inoculum requirement 5%, then aeration requirement 0.5 volume per minute. Finally, we can discuss recovery. At the end of the fermentation process, the broth is filtered. The filtration is done to remove the mycelium. Then we get filtrate. This filtrate is treated with N-butanol or methyl isobutyl ketone. Okay. It is for extracting the antibiotic from the filtrate. It is then absorbed to activated charcoal. It is for removing other impurities. Then the tetracycline is eluted and crystallized. So these are the steps involved in the recovery process. I hope all are understood this topic. Thank you.